Hi guys. So today let us discuss first step of cellular respiration, which is glycolysis. Glycolysis is common pathway in both aerobic and anaerobic respiration. So it has basically 10 steps. The first 5 steps are known as preparatory phase. The reason why it is known as preparatory phase, because during this phase preparation for gaining of final product, which is pyruvate, is done, in which during this phase the free energy of the intermediates of glycolysis is increased, and during preparatory phase ATP is used rather than being produced, and that ATP is used for increasing free energy of all these intermediates. So these 5 steps of glycolysis are known as preparatory phase. Once preparatory phase is over, like we get glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, then after that payoff phase starts. Now this is the phase. This is the phase in which ATP is actually produced. How, we will discuss right now. So let us start with glycolysis. Also, payoff phase also comprises 5 reactions. So first of all, we know that glucose is a 6-carbon compound. We already know it. So first of all, hexokinase, which is kinase, we know what they do. Kinases add phosphate group. So glucose is first converted into glucose 6-phosphate. That means at 6th position of carbon, 6th carbon atom of glucose, a phosphate group is attached by hexokinase. Hexo means hexose, because glucose is a 6-carbon compound. That's why it's a hexose sugar. So the enzyme which adds phosphate group to hexose sugar, which is glucose, is known as hexokinase. During this step, 1 ATP is used, which is converted into ADP, and phosphate group is added at the 6th position of glucose. We start with 1 molecule of glucose. Now this is an irreversible step. That means like whenever we see these kind of arrows, double arrows, this indicates that it is a reversible phase step. However, if this kind of arrow is used, then it is irreversible reaction. Irreversible in the sense, now if we want to reverse this step, we will have to provide some energy. It won't go on its own. But these kind of reactions, so because adding of phosphate is added via like whenever a phosphate is added to glucose and any molecule, it forms covalent bond, and covalent bond is very hard to break. That's why it is irreversible. Now glucose 6-phosphate is formed. Next step is catalyzed by phosphohexose isomerase. Now this enzyme is an isomerase. Isomer we already know. In isomers nothing happens. Only the compound, the molecule which has so many atoms, the atoms get rearranged. Nothing comes in, nothing goes out. That's why it is a reversible reaction. And since phosphate group is not being added, it's already added on glucose 6-phosphate. So now fructose 6-phosphate is formed. Isomerization through by the reaction known as isomerization, and the reaction is catalyzed by phosphohexose isomerase. Again, hexose, because it is still a 6-carbon compound, because fructose is also a 6-carbon compound, and glucose and fructose are more or less like they have same kind of atoms and same number of atoms. Only the 3D structure differs. So through isomerization, the atoms can be rearranged here and there. The third reaction will be catalyzed by fructokinase 1. Again, it is a kinase. Kinase, what kinases do? As I have already said that they add phosphate group to a molecule. So, and this enzyme is very important. During regulation also, this enzyme is very important. So we will talk about this enzyme again during regulation. So this is also an irreversible step, like here, because here one phosphate is being added again through covalent bond. So this reaction cannot be reversed easily on its own. We will have to provide energy in order to do that. That's why it is an irreversible reaction and we are using this kind of arrow. So in this reaction, because ATP is required, as I already said, it is a covalent bond. One phosphate is added, one more phosphate is added at first position. So earlier it was fructose 6-phosphate, now it becomes fructose 1,6-bisphosphate. Now the next reaction is catalyzed by one ATP is used. I think I have already talked about it. Next reaction is catalyzed by another enzyme known as aldolase. What aldolase do is it splits fructose 1,6-bisphosphate, like till here. These are all 6 carbon compounds. But once aldolase catalyzes its reaction, it acts upon fructose 1,6-bisphosphate. This 6 carbon compound splits into 2 molecules of 3 carbon atom each. Like glyceraldehyde 3-phosphate, we call it GAB also. And DHAP is also, dihydroxyacetone phosphate is also known as DHAP. These are the short forms. Now this is also a 3 carbon compound. That means now 6 carbon compound fructose 1,6-bisphosphate gets split into glyceraldehyde 3-phosphate. 3 carbon atoms are here and 3 carbon atoms are here. Dihydroxyacetone phosphate. Right? So the reaction is catalyzed by aldolase. Also, one more thing we should know about glycolysis. Dihydroxyacetone phosphate is formed before glyceraldehyde 3-phosphate, but later on it gets isomerized into glyceraldehyde 3-phosphate, like glucose 6-phosphate gets isomerized into fructose 6-phosphate. Again, this time the enzyme will be triose phosphate isomerase. Why we call it triose now? Because glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are 3 carbon compounds, not 6 carbon compound. Earlier we called it phosphohexose isomerase because this sugar had 6 carbon atoms. Now both the sugars have 3 carbon atoms, so we will call the enzyme as triose phosphate isomerase. Now that's all about preparatory phase, and during preparatory phase we use two ATPs basically. No ATP is being generated yet. Now this dihydroxyacetone phosphate gets isomerized into glyceraldehyde 3-phosphate. Now this 2 indicates that 2 molecules of glycer, because this the enzyme doesn't act on DHAP directly. DHAP gets converted into gets isomerized into glyceraldehyde 3-phosphate. Now what happens is glyceraldehyde 3-phosphate is further converted into 1,3-bisphosphoglycerate. 2 over here indicates that there are 2 molecules of glyceraldehyde 3-phosphate. Now here we will notice 2 things. 2 inorganic phosphates are coming here because glyceraldehyde 3-phosphate has phosphate group added at 3rd position. But here this enzyme glyceraldehyde 3-phosphate dehydrogenase, what it will do is it will add one phosphate group will be added at 1st position also. So now it becomes 1,3-bisphosphoglycerate. Why we are calling it dehydrogenase? It is an aldehyde. Now it is getting converted into a glyceric acid. Glycerate means glyceric acid. So that means here what is going on? When we convert aldehyde which has symbol CHO into carboxylic acid group, carboxylic acid which is known as glycerate or any other molecule like that. So it is COH, and this process is known as oxidation. And oxidation is removal of hydrogen. That's why we call this enzyme as dehydrogenase, because it is removing hydrogen and it is causing, and removal of hydrogen, as I already said in my previous video, that whenever hydrogen is removed, that means oxidation is going on. So that means glyceraldehyde 3-phosphate gets converted into 1,3-bis-phosphoglycerate. Here now we will notice NAD, two molecules of NAD plus, because here we have two molecules of glyceraldehyde 3-phosphate, so two molecules of NAD plus will be converted into 2 NADH plus H plus. Now that means we are getting high energy molecules. NADH is high energy molecule, and the energy is equivalent to around 2.5 ATP. One NADH gives energy equivalent to 2.5 ATP. Now this NADH will go to electron transport chain. We will discuss about electron transport chain later. And finally our goal is to get ATPs. So that means this step is very important. Also we can note that the phosphate, inorganic phosphate, is coming from, not from ATP. Usually we get from phosphate, in glycolysis we get phosphate group attached to the molecules through ATP. But here we are getting from an inorganic phosphate. This step must be noted down, because the examiners sometimes ask about it. Now, once we get 1,3-bis-phosphoglycerate, then this 1,3-bis-phosphoglycerate is converted into 3-phosphoglycerate. Now, and this step is in this step also, now it is being removed. One phosphate group is being removed from 1,3-bis-phosphoglycerate, and the phosphate is being transferred to ADP, so that 2 ATP could be generated. Now in contrast to the preparatory phase, in payoff phase, ATP, rather than getting used, now is being produced. So, and this kind of ATP production is known as substrate level phosphorylation. It is also being in GATE and in many exams they ask it, that during which step of cellular respiration we can get VC substrate level phosphorylation. So this is where we get ATP like this, and the process is known as substrate level phosphorylation, because from a substrate phosphate group is being transferred to the ADP, so that ATP so that we can get ATP. Right. So now we got 2 NADH plus H plus 2 direct ATP. Right. Now this step is also irreversible, because we are removing one phosphate, so obviously it requires energy to reverse the step. Now another step, now this 3-phosphoglycerate gets converted into 2-phosphoglycerate, and the enzyme is phosphoglycerate mutase. Now what mutases do is they only rearrange atoms, they rearrange phosphate group from one carbon atom to another carbon atom. So mutases, it is transferring the phosphate group from third position to the second position of the molecule. It was 3-phosphoglycerate, now it became 2-phosphoglycerate. So that means it is only shifting the phosphate group within the same molecule, and this kind of reaction is catalyzed by mutases. This step is reversible. So once we get 2-phosphoglycerate, now The next enzyme is enolase. Enolase converts phosphoenol 2-phosphoglycerate into phosphoenol pyruvate. It is adding carbonyl group to it. So that's why the final product is phosphoenol pyruvate, and 2 water molecules are removed from here, from this molecule. Again, we will see here substrate level phosphorylation. 2 ADP. This phosphoenol pyruvate will be further catalyzed by pyruvate kinase, and finally we will get pyruvate. So here also we will see substrate level phosphorylation. This step is also irreversible. One phosphate group is transferred from here to ADP to yield ATP. So finally, this is how we get pyruvate during glycolysis. Now this pyruvate, what will happen? What will be the fate of this pyruvate? In case the organism is anaerobic, then this pyruvate will undergo fermentation. But if the organism is aerobic, then this pyruvate will enter a PDH complex, where it will be decarboxylated oxidatively. So students, this is the overall reaction of glycolysis. That means by this reaction we can get to know what we invested during glycolysis and what was the output, what we gained. So glucose plus 2 ATP, which were used in preparatory phase, 2 NAD plus 4 ATP plus 2 inorganic phosphates. These were the investments. And what we get finally? What we got? 2 pyruvate molecules, 2 ADP, 2 NADH plus 2H plus 4 ATP plus 2H2. So these ATP will get cancelled out here. So what we will be left with? Glucose plus 2 NAD plus 2 ADP plus 2 inorganic phosphate, 2 pyruvates plus 2 NADH plus 2H plus 2 ADP plus 2 water molecules. It means finally we got 2 ATP and 2 NADH molecules during glycolysis. And now, in cellular respiration, our gain is to get ATP finally. NADH is a high energy molecule. Like it can give more energy than even ATP. But our body cannot directly use it. Our body can use ATP only. So like it's like a check. Like we go to bank, then we encash it. It is just like that. Because finally we need cash. So cash is like ATP. That means the goal is to get ATP from NADH. So this NADH is further reoxidized into NAD plus during oxidative phosphorylation during ETC, that means electron transport chain, so that we can get ATPs. One NADH molecule gives 2.5 equivalent ATP energy, and this is not the only step where we get NADH. Even more steps, the further steps of cellular respiration like TCA cycle also, it produces NADH. It also produces FADH2, which is a high energy molecule like NADH. It's just that it produces less energy as compared to NADH. It produces 1.5 ATP. So that was all about glycolysis. Now in next video we will talk about how pyruvate produced in during glycolysis is oxidatively decarboxylated.